In Experiment 3, we're going to build on what we know about titrations to do our first complexometric titration using zinc and EDTA. We're familiar with acid-base titrations from general chemistry. There, we mostly dealt with Bronsted-Lowry acid-base definitions, where an acid was a proton donor and a base was a proton acceptor. Here, we're introduced to a new type of acid-base chemistry that follows the flow of electrons instead of the flow of protons. In Lewis acid-base theory, the Lewis acid has room to accept electrons, while the base is bristling with spare electrons to share. Use the alliteration to your advantage. Acid, accept, bases are bristling with extras. Note that Lewis bases are also called ligands. Let's take a closer look at how Lewis acids and bases interact. In this generic reaction, we see the metal cation with room to accept electrons from the ligand, which is bristling with spare electrons to share. The product of this reaction is called a complex, which we'll discuss further in lecture. Ligands have the ability to donate one or several pairs of electrons. Halides, such as chloride, iodide, and bromide, are considered monodentate ligands because they can only donate one pair of electrons each to a Lewis acid. Ligands that are bigger, such as ethylene diamine or oxalate, can donate two lone pairs of electrons, making them bidentate. Multidentate ligands, such as EDTA, are big and flexible enough that they can wrap around a metal center, donating several lone pairs of electrons. When we have a ligand that can donate multiple lone pairs of electrons to a metal cation, this process is known as chelation. The word chelate originates from the Latin word chela, which means claw. Now, EDTA is a very useful chelating agent because we can adjust the number of binding sites it has available by adjusting the pH. For example, at pH 11, the acidic protons have been stripped away from each of the carboxylates, the COO- groups, providing 4 binding sites. Each nitrogen also has a lone pair of electrons to donate, giving 6 different locations for binding. This is accomplished by wrapping the EDTA ligand around manganese 2, as shown here, in an octahedral binding arrangement. Please pause the video and take a moment to identify each nitrogen and oxygen connected to our manganese metal center. Lots of complex ions form in nature that give rise to colors. For example, human blood is red due to the iron hemoglobin complex in our blood. Marine worms prefer to use an iron hemerythrin complex, causing their blood to appear purple, while other marine worms use a chlorochlorine iron complex that causes their blood to a characteristic green color. A related complex, hemocyanin, uses copper ions to transport oxygen in blood. The spiders, crustaceans, and octopuses that use this method of oxygen transport must be UK fans, right? Okay, moving right along. ERIOCHROME black tea is the indicator that we will be using for this experiment. Please pause the video and take notes on this slide. Please pause the video and take notes on this slide as well. Please pause the video and take notes on this slide as well. And again, for this one, please pause the video and take notes on this slide. As well as this one, please pause the video and take notes on this slide. Please pause the video and take notes on this final slide. Make sure that your lab report includes the average, the standard deviation, and the RSD values for all 3 trials, while making sure that you only have calculations for one trial. You do not need to have calculations for all 3 trials. Your lab report is 30 points.